Now that we reviewed the different parts of the cell and the different organelles and what they do, we could look at, which was the cell anatomy, we could actually look at the cell physiology and what's going on inside of that cell. There's actually several different major processes that happen within inside of the cell. The first one I want to talk about is membrane transport, which involves the cell membrane. The next one I want to talk about is cell division, like mitosis and meiosis, which happens involving the nucleus, as does DNA replication. And we're going to talk about protein synthesis, and I also want to talk about a process called cellular respiration. So let's go through these one at a time. The first one is membrane transport, which obviously involves the cell membrane. The main function of the cell membrane is for support and protection because it controls anything that enters or exits the cell. If we do a quick review, the cell membrane is made out of these special lipids called phospholipids. A phospholipid has 2 fatty acid tails and has a glycerol, but instead of the third fatty acid like triglycerides, they actually have a phosphate group coming off one end, which makes it polar. And the fact that it's polar means that one side of the phospholipid loves to be by water, because water's polar too, and the other side hates to be by water. It's hydrophobic. So it gives properties of this part of the phospholipid, the head, loving water, it is hydrophilic, and the fatty acid tails, they are hydrophobic. So when you get a bunch of these phospholipids together, they will spontaneously form what we know as a membrane in a double layer, where all the water-hating or hydrophobic tails are on the inside, and all the hydrophilic phosphate heads are on the outside or on the very inside of that, and that's where we get a cell membrane. We call it the phospholipid bilayer because it's made of a bunch of phospholipids, and bilayer means there's 2 layers, a top and a bottom layer. So hydrophobic and hydrophilic, because there's water on the outside of the cell and there's water on the inside of the cell. When we look at this membrane, we could also see in the gray the phospholipid bilayer, but these purple things, these are called membrane proteins. They act as little gates that allow big stuff to go in and out of cells. And then we have these they're called carbohydrate chains. They're sugars that receive chemical signals telling these membrane protein gates or channels whether or not they should open or close. And so it all kind of works together like that. There's 2 major ways to move things in or out of a cell. The first major way to move things is through passive transport, and the second way is through active transport. Passive transport, if you are passive, you don't really exert much energy, so this is a process where you don't need any energy or ATP to move things from one side to the next. There's actually a couple different examples of this. You'll know the easiest one is diffusion, but there's also one called facilitated diffusion, and there's one called osmosis as well. The other way that you can move things across a cell is by active transport, which does require energy. And so the main types of active transport they're called protein pumps, or you could do endo or exocytosis. The passive transport also usually goes from where there's more to less, whereas active transport pumps things from where there's less to more. So we call this going down the gradient from a high to low concentration. Whereas active transport, if we look at these we'll look at the square molecules, there's not so many here, so we're going from a low concentration to where there's more from a high concentration. So passive is no energy, it goes from high to low concentration. Active transport requires energy, and it pumps from a low to a high concentration or against a gradient. Let's look at passive transport first. One thing with passive transport, no energy, and you go from high to low. There's 3 different types of passive transport. Simple diffusion, where molecules just go right through the phospholipid membrane. Molecules like gases, like oxygen and even carbon dioxide, they could fit right through with no problem. Sometimes things can't fit right through, and so they need to go through an open gate, and we call that gate facilitated diffusion. Facilitate means to help, and so you need a gate to help larger molecules or polar molecules move from high to low concentration. And then we have another one where you move water from a high to low concentration. And so osmosis is the third type of passive transport, and these are low water molecules. Water needs a special gate to go through called an aquaporin, and it goes from where there's more water to where there's less water. And when you look at osmosis, and you talk about the movement of water from where there's more water to less water, cells rely on osmotic pressures. And we have 3 different types of tonicity based on this. We have hypertonic, isotonic, and hypotonic. And so just a quick review is, where is the water moving in relation to the cell? And so in this black or dark blue area, that's the solution that these red blood cells are in. And if for some reason the cells are in a hypertonic, hyper means above concentration, tonic is concentration, hyper is above concentration. If there's the solution out here is above concentration, so if there's a whole lot of salt, NaCl, out here, more so than they are inside of the cell, then the water will have to rush to where there's a higher concentration. So water will leave the cell and it will shrivel up and crenate. Over here, isotonic is where your body cells inside of you like to be most of the time. They like to have a same concentration on the inside and out. That means that the amount of salt, for example, or sugar, or whatever compound you're talking about, is equal on the outside versus the inside of the cell, which means that you're going to have some water always moving in and some water always moving out. That's isotonic. And then there's hypotonic. Hypo means below strength, and so that means that whatever's surrounding your cells is below strength in comparison to what's inside of your cell. That means that there's less salt on the outside of your cells than the inside of your cells. So water will move to where there's more stuff. So water will go to where there's more salt, which means water will rush into the cells, and that will cause the cells to swell and eventually burst or undergo cytolysis, which is not a good thing either. So your body always wants to be in an isotonic environment. Your body also does this. When does your body do this? It does it regulating how your kidneys work. So your kidneys are able to filter out the right amount of water because they're regulating the tonicity of the different cells and in your urinary system. So that's an important thing as well. Let's look at active transport. Active transport is when you do need energy because it is active, and so you'll see ATP being available, and you're going to pump things from where there's less to more. So these hydrogen ions are being pumped from where there's less to more. So low to high concentration. And why do you want this? It's because that's how your muscles and nerve cell works. They have to keep a certain pH on one side of the cell in comparison to the next side of the cell. And eventually it allows for cell communication through an electrical impulse. So this is exactly how electrolytes keep your cells functioning, because you have a certain electrical current going across your cells and your cells communicating from one cell to the next. The next type of active transport is called endocytosis. Endocytosis. And that's just simply stuff that forms in a vesicle. The vesicle surrounds it and takes the stuff inside of the cell. The opposite is called exocytosis, where materials inside of a vesicle will fuse with the cell membrane and be released. And then over here, this is just showing endocytosis and exocytosis always occurring while cells doing its normal functioning. Next thing I want to talk about is cell division. There's actually 3 types of cell division. One that's done with bacteria. Bacteria will cell divide to unicellular organisms, even protists. The only reason that they would undergo cell division is to make more of themselves, which is reproductive. And they do it usually through a process called binary fission, where they just split into 2. However, this would be called asexual reproduction as well. However, we do cell division in 1 of 2 ways, through mitosis or meiosis. And the reasons for those, if you're made of more than one cell, is because you're either growing, you're either maintaining cells as old cells die and you need to replace them, or you need to repair an injured tissue or organ. And when we do meiosis, we actually need cell division to make our sex cells or our gametes through a process called meiosis. So I'm going to talk about mainly mitosis and meiosis as a review real quick. Looking at the cell cycle, we have different phases of the cell cycle. This is how a cell grows and divides. So when a cell is first produced, the cell cycle always starts at this part called G1. A cell will grow, and it will grow and it will grow. Eventually it will reach something called a checkpoint, where it needs to make a decision on whether or not it's going to exit the cell cycle and just kind of do its normal cell work, or whether it's going to get a signal saying, yeah, we need more of you, so you're going to have to divide and make some more. And so once it passes this checkpoint, it is required to go to the next part of the cell cycle, which is called S phase. S phase is where DNA will replicate and make more of itself. And then after S phase, you'll go into G2 phase, where any last minute preparations and kind of it checks to make sure everything's good to go. And then you'll finally move into the division part of the cell cycle. The cell cycle is divided into two major parts: interphase, which includes G1S and G2, and M phase, which stands for mitotic phase, or the cell division phase, which includes mitosis and cytokinesis. And mitosis is broken down further into prophase, metaphase, anaphase, and telophase. When you look at S phase and DNA replication, so after G1 and you go into G2, you have something that happens in S phase called DNA replication. If you have one cell and you need to make two cells, you need twice as much of everything that you had, including your DNA. So you'll do a process called DNA replication, where you take your original strand of DNA, you will break the hydrogen bonds that hold the two sides together, and then you will, you know that A's and T's and C's and G's go together in DNA, so they will be replaced with their complementary pair to make two new exact copies. And a couple terms I want to refresh your memory on is complementary. Complementary, all it means is that A is complementary to T and C is complementary to G. They go with each other. And that's how you know what you could fill in to make an exact copy of what used to be there. Another term I want you to know is semi-conservative. Conservative means that you, in the end, you get two exact copies, but they are semi-conservative. Semi-conservative means that you have half the original, so half of the old stuff, and then you get half new. So semi means half. Half of it is conserved and half of it is new, semi-conservative DNA that's made. And the last thing I want to talk to you about is anti-parallel. DNA is anti-parallel, which means that one side of the DNA, because it's double-stranded, is running in one direction, but the other side runs in opposite direction. Sort of like a street where traffic could only flow in one direction. And then the A's and T's and C's and G's are matched up like this. So really one side is upside down to the next. So anti-parallel, you end with a phosphate on one side, but the opposite side is a phosphate, and over here we have a sugar, and down here we end with a sugar. So that is anti-parallel, and it's very important for when the DNA is replicated, which I'm not going to get into. There are a lot of enzymes that help regulate this process. And so I'm not going to talk about this, but if you took a more advanced biology course in your previous classes, you'll remember all these enzymes like DNA helicase, DNA polymerase, DNA ligase that actually help replenish and make the new semi-conservative strands of DNA in certain directions as well. Now, when you go back to the cell cycle, please remember that that was interphase, G1, S, and G2. And over here we have M phase, which consists of mitosis and cytokinesis. So let's look at mitosis and cytokinesis. Mitosis, just a quick review. You start in interphase. Interphase is when DNA is all scrambled up in chromatin form. That means it's not coiled up. And you have the nucleus and all the organelles and everything's there. But then when it's told to undergo cell division, it will have to actually separate the DNA that was copied during DNA replication. So what's going to happen during prophase is that this is too messy. It could get tangled up. So you need to kind of pack up your DNA into different tiny structures called chromosomes. And that's why they're this X shape, because they're sister chromatid form that's held together by a centromere. You could see it better down here. So chromatin is sort of like spaghetti all wound up. Your DNA will kind of spiral up like this into the chromosomes. They're held together by the centromere. And what you're going to do in this process is separate the two identical sister chromatids. This side is identical to that side. And that's what happened during DNA replication. So up over here, prophase, they kind of start to coil up. Prometaphase, which is an in-between phase between the two, is when you have your duplicated centrioles, and then they move to opposite ends. You have spindle fibers that connect to the centromeres in order to line them up in metaphase. In anaphase, they get yanked away from their partner or their sister chromatid, and they end up at the opposite poles of the cell. The nucleus will reform around those chromosomes. They start to relax back into chromatin. The spindle fibers that separated them will go away, and you start to do a process called cytokinesis, where you split the two cells equally. And so you could actually see that process happening here. And hopefully that was a very quick review of the entire process of mitosis. Here's a comparison between mitosis and meiosis. Mitosis, pretty much you start with a cell that in this case has 6 chromosomes instead of 46, and we call this diploid because it's 2N or diploid. They will actually copy each other, prophase, metaphase, and line up in the middle. The spindle fibers pull them apart in anaphase, and then telophase, they will start to unwind back into chromatin, but you get 2 new identical cells that are still diploid. They have the same amount of whatever they started with at the beginning. It's a cloning process, mitosis is. Meiosis is a process of making gametes. That's the only time it is done. So this is for all cells except for gametes, which is done by meiosis. Mitosis, copy or clone your cells. Meiosis, make your gametes or sex cells for reproduction. The main difference is that if you start with your 6 chromosomes, they will line up with their homologous pair. So mom's chromosome number 1 will line up with dad's chromosome number 1, and then they actually do this process called crossing over where they swap genetic material to mix up the traits, and then they line up with their partners in metaphase, and then they're separated once. So they go through a first division, but then immediately right after that, they're separated from their sister chromatid as well. And so in the end, you get cells that are haploid, which means they have half the number of whatever you started with, because gametes are haploid. They only have, in our case, they have 23 chromosomes, where all of our other cells have 46 chromosomes. And so that is mitosis versus meiosis. One thing I want to point out real quick is that the aging process really comes down to the chromosomes and the division process. The reason that we get older and we don't maintain our cells as quickly and as good enough is because the whole process slows down as we get older. Our chromosomes have ends called telomeres. The part right here is called a centromere, but the end is called a telomere. These get shorter through every cell division, and the shorter they get, the slower your cells divide. And the slower your cells divide promotes the aging process. And there's a lot of different research going on with that, and we could talk forever on that process of aging. So as you age and injury increases, healing will decrease because of the telomeres getting shorter. The next process I want to talk about is protein synthesis. Protein synthesis is the process of making proteins. So the DNA copies and makes a smaller copy called RNA in a process called transcription. And then your RNA goes out into your cell, ribosomes, and they will translate this code, this genetic code, into the actual protein of what it's supposed to be in. So DNA to RNA to protein. And if you look at the nucleus, your DNA is found in your nucleus. Your DNA is too important, so it cannot fit outside these nuclear pores. So it will make miniature copies called mRNA, or just RNA. There's 3 different types of RNA. The RNA is able to leave through the nuclear pore. It goes to the ribosomes. The ribosomes latch onto it and read it, and then it reads it in a code that tells it what these purple circles are. Those are the amino acids, and it makes a chain of amino acids, which is called a polypeptide. The polypeptides will fold up into a certain protein, which is functional. And this is a somewhat more detailed example of it. This actually shows the red DNA making the yellow RNA leaves the cell, and then, so that's transcription, DNA to RNA. Translation is happening in the cytoplasm where RNA is read by the ribosome to actually make the protein. Now, how does that happen? You may remember this fun wheel that we used to read from the inside out. And so your genetic code is A's, G's, U's, and C's. Remember that the U is replaced from the T during the transcription process. But your ribosome will read it in code, 3 letters at a time. We call these codons. And each codon represents an amino acid. So if we read AUG, AUG, that's methionine, that's why it says met. And then it'll read ACU, ACU, that's threonine, and that's why it's THR. And so it reads it down the RNA, and then eventually this forms a polypeptide that will fold up into a functional protein. And that's a process of protein synthesis. And so then after we make the proteins at the ribosomes, if we look at the cell, proteins are made here on the rough ER by the ribosomes. Those proteins get shipped out in vesicles to the Golgi. The Golgi packages them up and ships them out to where they need to go. And so the whole process works how it should inside of the cell. The next process is cellular respiration. Cellular respiration is a process of making our cells energy, which is ATP. So this is our cellular energy. But we need glucose in order to make the ATP. Glucose it's too big of a molecule to use for our energy source. So we use this chemical compound called adenosine triphosphate. You eat some food, mainly glucose, you breathe in some oxygen. Inside of your mitochondria, there's special chemical reactions that happen in which you make the ATP, but you also get rid of waste products that you do not need in the end. And so here's a picture of what ATP looks like. Adenosine, which is the adenine and a ribose sugar, and then there's 3 phosphates. Now, your body uses the energy when it breaks off this 3rd phosphate right here. When it breaks it off, it releases a whole lot of energy that your body's able to use to do all of its processes, and that's why this molecule is so incredibly special. Looking at the mitochondria, here's the mitochondria where cellular respiration actually occurs, and there's different parts of it, but overall what will happen, the glucose will come into the cell, so this is actually the cytoplasm or the cytosol of the cell, will enter it, and then it will be quickly converted in a process called glycolysis into a molecule called pyruvate, and then pyruvate has one of 2 options. If there's no oxygen present, we do something called anaerobic respiration, which is also known as fermentation, and that's where you do lactic acid fermentation, where if you ever run and feel burning in your muscles, that's the lactic acid building up. We don't do alcoholic fermentation, but bacteria and yeast do that. This pyruvate, which is a different form of glucose, could also, if oxygen is present, we do aerobic cellular respiration, in which the pyruvate enters the mitochondria, and then it goes through some different steps like Krebs cycle and then the electron transport chain in order to make a whole lot of ATP. And that's a general overview of the process. If we look at a more detailed view of the process, it depends on what biology course you took. This may be familiar to you and kind of a review, but if you just took college biology, you probably didn't even see this before. So I'm going to not going to make you responsible for knowing all the steps, but I want you to understand that cellular respiration is more than breaking down glucose and all of a sudden there's ATP. There's a long, complicated chemical reaction process that's involved to make them. And so it all starts with eating glucose. So here's your glucose. It's broken down into this molecule, sugar molecule called pyruvate, in a step called glycolysis, which does not even happen in your mitochondria. It makes a little ATP, but not enough. And then the pyruvate, if there's O2 present, it will be converted into a new substance that will enter step 3 called the Krebs cycle. The Krebs cycle is also responsible for making some for making some ATP. This conversion and this step called the Krebs cycle also makes all the carbon dioxide that you breathe out. So when you breathe out right now, that's the Krebs cycle talking, and the conversion reaction. It's all that stuff that you breathed out is from inside your mitochondria as a waste product. After the Krebs cycle, I mean, we only have 4 ATP, which is not that much. Every glucose molecule can make approximately 32, 34 ATP, and so we have to get up to that number, and 4 is not cutting it. And so the Krebs cycle makes some molecules called electron carriers, as does glycolysis. An electron carrier carries electrons. Electrons are important because the electrons have to go to the electron transport chain. Electrons are important for making a whole lot of ATP in some complicated processes called chemiosmosis and oxidative phosphorylation. But electron transport is able to convert the rest of that into lots and lots of ATP. And so in the end, we also need some water to do this, which is why water is important, but we also give off some oxygen as, this is backwards, I'm bad. Let's reverse this. The arrow should be pointing this way. Water is given off as a waste product, but oxygen, you breathe in oxygen because you need it, so oxygen should be going this way instead. You breathe in oxygen because you need it in order to make your ATP. And this is, again, a complicated overview of the entire process, but cellular respiration is more complex than just looking at a chemical equation like this. So keep that in mind as well. Now, when we look at the foods that we eat and how we break it down, I know that we always talk about glucose being the main source of energy, but that doesn't mean it's the only source of energy. Glucose is the primary source of energy because it's the easiest to convert through the steps into the ATP. But if we don't have the sugars or carbs necessary, we use fats, and in the last case scenario, we do use proteins for energy sources as well. But first and foremost, we use carbs, our backup are fats, and we don't like to use proteins, but we will if it's dire, if it's a life and death situation. All these fats can be converted into the 4 different steps of the Krebs cycle to make our body's ATP. All right, and I hope that was helpful to you. That was pretty much almost 2/4 of biology in, less than a half an hour. So I hope that made sense to you and it was a good review, and you asked me questions if you need more clarification on anything. All right, bye.